Learning live webinar. We have session one, which is Introduction to Western Blotting. Let me put on my laser pointers. I'm Kailing. I'm the Regional Field Application Scientist from Azure Biosystems. We have 4 important agenda today. The first is principle of Western blot. Second, the antibody detection with chemiluminescence and fluorescence method. Imaging Western blots, and finally, Western blot normalization. Just a brief introduction about our company. We have Azure Biosystems. Azure Biosystems is founded based on innovation. It is co-founded by Arnold Shifji and Dr. Diping Che. Arnold is our CEO, and Dr. Diping is our executive or VP of R&D, where he had led all the design and development of our imaging system and a lot others of instruments. Azure Biosystems is founded in 2013. We're based in Dublin, California, in the United States, very near to Silicon Valley, where we designed, developed, and manufactured all our products there. The core members of Azure Biosystems are coming from Alpha Innotech, which then acquired by Sulfur Assayers and become the label that we are familiar today, which is Protein Simple. So in 2013, we have Azure Biosystems. We have been aiming to give you raw data and the data that you can rely on using simple workflows that you can trust. At the moment, we have 3 key important instruments. Firstly, Sapphire Biomolecular Imager, Azure Imaging System, and also Azure Cielo Real-Time PCR Systems. Right, let me go straight to the topics. This year, the year of 2021, is the 41 years of Western blotting. So this figure is taken from the Journal of Proteomics, published back in 2020, last year. It shows the proportion of 5 different techniques in all protein-related publication, estimated by the PubMed mentions in text, word, and titles. So we can see that even though the title of the Western blot in the journal is reducing over the years, but this method has been consistently high in proportion compared to other techniques that to quantitate protein expression or to analyze it. So this shows that Western blot is the essential and widely used analytical techniques in protein studies. One thing is, Western blotting is universal. We have 2 major questions when you're running Western blot. Firstly, qualitative question, and secondly, quantitative question. So what is qualitative question? Qualitative question means yes or no. Do I see the protein presence or not? Quantitative means what is the measure to the changes or the expressions level when comparing it to another controls or another group. This is a simplified flowchart of Western blotting workflows. It starts with electrophoresis to separate the protein loaded according to the molecular size, then transfer the proteins from the gel to a membrane. Next, incubate the membrane with a blocking reagent to reduce the background noises. After that will be the primary antibody incubation. Then from there, you can proceed to choose either to do chemiluminescence or fluorescence detection. Right, let me go to the principles of Western blotting. The principle of Western blotting on the first step, which is gel electrophoresis. The idea of this step is to physically separating the proteins from one and another across the gel matrix with gel electrophoresis. How we do is you load a protein sample mixed with a loading buffer, loaded them into the gel, then put them in an electrical current that is applied to the buffer or gel system. We can use SDS page gel, resolving gel, which is quite a commonly used gel to separate them, and or we can say resolving the proteins based on the gel pore size. We have Lamley discontinuous gel, allowing you to separate for sharper and more defined bands, and also gradient gel. Those are commercially available, ranging from 4% to let's say 20%, allowing you to separate or resolve multiple proteins that's spanning a wide range of molecular weight. Normally, standard SDS page gel use alkaline buffer such as tris glycine for more mid-size protein resolution. There are two choices in terms of membrane in the transfer step, where the transfer step is the aim is after you separating the proteins according to their molecular weight, you want to transfer this protein from the gel on a membrane. So we can do the next steps. There are two choices here, nitrocellulose versus PVDF membrane. Nitrocellulose is more cost-effective than PVDF membrane and lower background for chemiluminescence detection. It doesn't need any activation, but it is fairly fragile and it cannot be used if you're doing fluorescent detection method. PVDF, on the other hand, is much more durable, suitable for both Chemi and fluorescence detection. It requires methanol to activate the membrane before you use. In terms of fluorescence detection, low fluorescence PVDF membrane is the best option if you want to do the transfer step. Right, transfer we have generally divided to two types. First is wet transfer, where you soak the whole sandwich into a tank full of buffer. Another one is semi-dry transfer, where you have the blot and the gels sandwiched between few layers of at both sides, few layers of buffered soak or buffer wet filter papers and carrier. So these are the few advantages and disadvantages of wet transfer and semi-dry transfer. Wet transfer has been used conventionally because it can provide you the flexibility in terms of adjusting the transfer condition, and it allows you to do multiple transfer buffer options, supporting a transfer at a broader molecular weight, and compatible if you want to transfer bigger proteins and allow you to run it for a longer time at a lower voltage. And it can be used for quantitative Westerns. However, there are a few disadvantages as well. Wet transfer, the heating can be interfering with the transfer. The cooling mechanism need the cold space, room space, or cooling system. And also you need more volume, the transfer buffer volume to fill up the tank. Right, for semi-dry transfer, the best thing is it's fast. And you can also use the discontinuous gel to optimize the transfer of the proteins ranging from 4% to 20%. You don't need a lot of buffer, unlike wet transfer, and it is easy to set up the sandwich. It is also good if you want to transfer multiple gels at the same time. But there are a few drawbacks as well. For example, due to the high intensity of the electrical charge, you might cause a low molecular weight protein to over-migrate passing the membrane, thus you're losing the low molecular weight membrane. And it has also high difficulties in transferring high molecular weight proteins that is more than 120 kilodaltons. And it is not very recommended for quantitative Western. And finally, the gel can be dried out for a longer time. So these are the options or components that you can use to set up your or make the transfer buffer. We have trees and so on, buffer, alcohol, detergent, and trees triazine for their own purposes. Next thing is voltage and time. So transfer time and voltage setting should be optimized for each transfer due to unless you have optimized everything, let's say how you do the sandwich operator and also the buffer volume and so on and so forth. So in general, the longer the transfer time it is, you should use a lower current in the wet transfer. But if you're using semi-dry system, it's best to do a higher current transfer that gives you a shorter transfer time. Because when proteins are generally transferred more rapidly at higher voltage, the transfer efficiency is not always consistent. So this insufficient current and also time may result in incomplete transfer. That means whatever you have on the gel will not be completely transferred on the membrane. And while high current and lengthy transfer time may also cause you to lose the protein via the transfer that is over transferred of passing the membrane. Now, this blocking step at the Western blotting protocol, the blocking allows you to remove non-specific binding of antibodies that can result in higher background signals when you image the blot, thus reducing the detection sensitivity. The idea is to block the membrane after you transfer it. And so these are the few components to use. The first is buffered salt solution. You can use either TBS or PBS. Another thing you should be beware of is PBS can interfere with any alkaline phosphatase-based detection method or phosphor-specific antibodies. So if you are trying to, let's say, you're trying to detect a phosphor-related protein, it's best to use trace buffer. Detergent, low concentration of mild non-ionic detergents such as Twin 20 is recommended to use when you're preparing the blocking buffer. The purpose is to add and just to prevent the microbial growth of your blocking buffer. And just to make sure that you will not leave any residual during your detection, you have to make sure that the detergent is completely solubilized just to prevent any artifacts. Another key thing is if you're doing fluorescent detection method, we should not use detergent as a blocking agent. Continuing, another agent that we can use as a blocking buffer is protein. You can use non-fat dry milk, which is economical, easy easy to prepare, and is very efficient, but it's not very suitable for phosphor-specific antibodies. You can also use serum. It's less favorable because it gives you higher background. Another is bovine serum albumin. This is good for phosphor-specific antibody, but you have to be wary as well if you're doing tyrosine phosphorylated protein content. And it cannot be used with anti-lactin antibodies as well. Right, you have another option in terms of blocking buffer, protein-free. There's option for you to use water-soluble polymer polyvinyl pyrrolidone, which is commonly used in southern blot. These type of buffer can be used for Western blot as well. Right. What's the condition for blocking? Generally, the blocking efficiency is depending on the time and temperature. Of course, 1 hour at room temperature blocking is sufficient. But if you want to block for longer hour, more than 2 hours, it's recommended to block the membrane in 4 degrees Celsius. Next is membrane incubation with antibodies. So in terms of primary antibodies, we have two types of detection. First is direct detection. Here, this is direct detection. And you have indirect detection. So direct detection means you have a primary antibody attached with a detector label, which is either enzyme or phosphors and so on and so forth. So this is simple and straightforward. You just need to incubate this primary conjugated antibody, and that's it. For indirect, you have 2 different primers need to use. Primary antibody, secondary antibody, and also a detector molecules, example, easier substrate or anything, just to recognize a secondary antibody. And for indirect detection is much more popular and much more sensitive. It amplifies the signals, also highly cost effective. In terms of primary antibody, we have two different options as well, polyclonal and monoclonal. So polyclonal is just a mix of an individual antibodies that recognize different regions of the same antigen. It gives you a wide range of binding specification, but it also can lead to higher background noise. For monoclonal antibody, it recognizes all these antibody in the same solution will be recognizing the same epitope on the antigens. It gives you less robust signals, but it is highly sensitive. It also gives you lesser background noise. There you go for your primary antibody. Now, primary antibody. Now we are talking about secondary antibody. In terms of secondary antibody, we have the choices based on species and class type. There are a few species used in producing the secondary antibody, rabbit, horse, mouse, goat, donkey, and so on and so forth. There are different types as well. The isotype is depending on the type of heavy and light chains that makes up the antibody. So I will not go into detail because this is very much into the depth of immunology. Next is antibody conjugate. We have enzymatic labels such as alkaline phosphatase and horse radish peroxidase HRP for chemiluminescence detection. Fluorescent label for fluorescent detections like CyDyes and GFP, RFP. Others such as biotin labeled antibodies for more sensitive detection for low abundant proteins. Right. Next, we go into details about chemiluminescence and fluorescent Western blotting at the detection part. Right. Chemiluminescence is Western blotting is a one signal. It's best for one signal and one protein. In chemiluminescence detection, the antigen and primary antibody complex is bounded by secondary antibodies that's conjugated with the enzyme. Here is a horse radish peroxidase HRP or EPI, alkaline phosphatase. This enzyme will later catalyze a reaction by using the substrate that will generate light in the presence of a luminescent substrate, I said before. And then the light can be detected either using X-ray film or CCD-based imaging system. Here, one thing we have to be careful is the chemiluminescence signals relies on this enzymatic substrate reaction. So the amount of light or emitted light of the signals is subject to a lot of variation, such as pH, temperature, substrate concentration, and enzyme concentration. So due to the nature of this enzymatic substrate reaction, the signal or band intensity increases greatly over time and also based on depending on your protein abundance. But this is quite problematic if you have highly expressed protein that will saturate, the first thing, saturating your membrane. Second is saturating your detector. First, we need to be careful on the ECO substrate that you use because ECO substrate is heavily affected by all these reaction kinetics factors. Here we have for comparison of 4 different ECO substrates. We have Armacham, Armacham Plus, Super Signal, and also Radiance from Azure Biosystems. Here I want to tell you that all these ECL substrates are good. They are working fine to a certain limit. You can see on the graph, the figure over here, the graph. So in Western blot, to quantitate it accurately, there's one thing we have to be careful is something we call as signal linearity. That means whatever your protein load is, the signal must be directly proportional to the protein amount. You can see all of these substrate, chemiluminescent substrates, they are good. They have give you good linear signal linearity. But if you look closely, most of them are not longer linear above 1,000 picogram of protein load, such as these indicated in the square balls and triangular. So what it means that if you have protein load that past 1,000 picogram, in a single load, this ECL substrate might not be applicable or useful for you to quantitate the protein expression because it is not longer linear and it is not longer quantified or qualified for quantitation, as it is not longer representing the actual protein amounts that you're looking for. Right. So chemiluminescence have been used widely. That is something that we cannot deny. Of course, it provides a lot of advantages. First thing, it is super sensitive. You can detect up to femtogram levels. The method is easy. It has a familiar chemistry that we have been familiar with, and the techniques is compatible with X-ray film, or you can use digital imaging system to detect the signals. This technique is very good if you want to ask in terms of answering the question, is my protein there or not? Like, I want to see whether the protein is present or not. But it is not very good in addressing the questions such as how much my proteins is present relative to another proteins, and how much of I control for sample loading inconsistencies. The chemiluminescence reaction emit light over a broad wavelength. Thus, we cannot use it to distinguish signals from different proteins on the same plot. So instead, the protein load must be properly separated during the gel electrophoresis step, the first step. So we can separating the controls from your protein of interest. And also due to this, I'll say feature, since it cannot distinguish different proteins, it is difficult. Chemiluminescence is difficult to visualize multiple bands at the same time on the same plot, because since the bands are most likely overlap, stacking on one another, so these overlapping bands can also impact the detection of your, let's say, housekeeping protein or your controls and loading control and so on. So unless these controls are separated properly from the protein, your protein of interest, there are two ways in doing this. Either you separate it properly, but if you cannot separate it properly, you either do a strip and reprobing step, and also either you do it on another gel just to hoping that it will serve as a proper control. But as a matter of fact, this actually making chemiluminescence detection not longer quantitative, because you see the controls are at a separate blot, and then or you strip and reprobe. There are evidence and publications showing that when you do stripping and reprobe, you will lose certain protein amounts, which no longer making the protein load the same anymore. So furthermore, that makes it chemiluminescence at best give you semi-quantitative power, right? And finally, the X-ray film you use in the chemiluminescence detection method has also limited dynamic range and also saturation issue. Right, next we go to fluorescence Western blotting. Fluorescent Western blot is fairly easy. It uses secondary antibodies that has direct conjugated with four dyes. So these four for Western blot can be chosen based on their specific excitation and emission wavelength. So as you can see, the excitation and emission wavelengths, the curves are separated. This is what we call as spectral separation. Because of this spectral separation between, let's say here, we have two different fluorophores here, it allows you to do multiplexing and detecting multiple proteins on the same plot at the same time for faster and more efficient studies. So first, one of the biggest advantages of fluorescence detection is when comparing with chemiluminescence is the ability to use more than one antibody per assay per image. The fluorescence Westerns are typically using digital imager rather than X-ray film, but it allows us to enjoy the newer generation of imaging system that contains much higher or level of sophisticated camera that normally gives you broader dynamic range than film. Here I want to highlight that fluorescent signals will give you stable and linear light production, and because due to the nature of these fluorochromes are not enzyme like HRP, thus giving you a linear fit that also gives you the quantitation power in Western blotting. Right, so Fluorescent Western blot gives you a lot of advantages. The first thing is definitely multiplexing capability and increase your quantitative accuracy, right? And can you visualize allows, let me go back to the multiplexing capability. It allows you to visualize proteins that cannot be separated properly in the gel electrophoresis step. For example, in post-translational modification study, for example, the total stat 1 protein and phosphor stat 1 protein, where both of them are not much difference in terms of molecular weight. Thus, you cannot separate them in the gel electrophoresis step and on the blot as well. So in this case, fluorescent Western blot is the best options to detect these two proteins at the same time or on the same blot. So you do not need to strip and reprop by having another blot that just to stain for the phosphor related stat 1. Okay. Unlike the chemiluminescence Western, which are limited by the kinetics and also enzymatic nature, the amount of light emitted from the fluorophores is highly consistent and directly proportional to the amount of proteins on the membrane. So what this means is if you have low abundance of proteins, then the fluorescent signal will be lower. If you have a high amount of proteins, the fluorescent signal will be higher. But when you image it across the time, the excitation time, the signal will remain the same. Unlike chemiluminescence, you have increase of a signal that will saturate the detectors. Thus, you will see a signal saturation indicator in the chemiluminescence capture. Right. So this consistency means that fluorescent detection can provide truly quantitative analysis of your protein of interest. One of the advantages is these fluorescent styles or labels are relatively stable. Your blot can be archived and imaged months after the initial experiment, as long as the precautions are taken to avoid photo bleaching of these fluorophores. Well, fluorescence detections is the best choice for quantitation. It can also greatly accelerate your workflow for analyzing multiple proteins on the same blot. This method has also a few drawbacks. As you can see here, it can be less sensitive than chemiluminescence. And another issue is the reagents and membrane can be autofluorescent, thus increasing the backgrounds. So the time taken to complete a Western blot workflow, when we compare between chemiluminescence and fluorescence, you can see here, it's actually, if you're performing fluorescence Western blot, the time taken will reduce to down to 3 hours, because there is no need to strip, wash, blocking primary antibody, secondary antibody again and again. Okay, so we can complete the whole Western blotting protocol if you're doing fluorescent detection within 3 hours. And you might wonder, in our common sense, we thought that fluorescence Western blot is quite expensive. In fact, it is way cheaper than chemiluminescence, especially if you are using X-ray film. Here we break down the cost of each key consumables in US dollar. In fluorescence Western blot, we only need to load a very minute amount of the fluorescence protein marker, due to the high and stable signal intensity. And yes, the secondary antibody can be slightly more sensitive than the HRP antibodies we use in chemiluminescence. But since there's no need for extra film and also ECO substrate, Fluorescent Western blot can cost you as low as 72 cents, where chemiluminescence can cost you up to $50. So if you, the price might be different in terms of regions in the world, but if you calculate it comparing between chemiluminescence and fluorescence, the reduction of the cost is having a lot of evidence. Right. Next is which is right for you? Chemiluminescence and fluorescence detection. When you're doing Western blotting, if you need to choose between these two, then you need to do chemiluminescence when you just want to detect, firstly, a single protein. Next, you just want to check whether the protein is there or not. Thirdly, you want to measure antibody response, or this is the step after protein purification. And also finally, if you want to detect low abundance of the protein. These advantages, or I would say these criteria, is all due to the nature of the chemiluminescent signals, which is kinetic and enzymatic substrate nature, thus giving you higher sensitivity, but it has other drawbacks as well. For fluorescent Western blot, you should use fluorescent Western blotting when you want to detect multiple proteins simultaneously on the same blot, and especially if you're doing post-translational modification study, where you want to in terms of quantitative Western blot, if you want to quantitate the same blot loading control for more consistency and also have inlay normalization. And finally, fluorescent Western blot give you the most quantitative power compared to chemiluminescence Westerns. So this is all due to the spectral separation nature of the fluorophores, allowing you to do multiplex capability. It is indeed much more cost efficient, also giving you direct correlation between signal intensity and protein abundance. But one, I'll say minor drawbacks is it can only be imaged using digital imaging. Here's a figure or flowchart that allows you to make decision either you want to do Chemi or fluorescent Western blot. First thing, the most important thing, do you have access to an digital imager? If no, then the answer is chemiluminescence. If yes, then there are a few other questions that you need to ask. First is, do you need to detect multiple proteins on the same blot? Are they at a similar sizes? And finally, do you need precise quantitation? If these are the questions that you need to be answered, then fluorescent Western blot is the way to go. However, if you do not have a digital imager and you need to quantitate or image a very low level of protein, then chemiluminescence is the way method should be used. Right, next to imaging Western blot, what should we do with it? Here we're talking about chemiluminescence Western. One thing I want to highlight here is every detector, we're talking about either using X-ray film or digital imaging, have their own intrinsic dynamic range. We always say wide dynamic range. What is wide dynamic range? The wide dynamic range means the ability of a detector to capture both very low and very high signal intensity in the same image without saturation. For quantitation, we promote or we recommend user to do digital imaging because as you can see here, film at both giving you 1.5 optical density, but using CCD chips, here there's example from Azure 600 imager, you can gives you dynamic range up to 4.8 optical density. Right. Now this brings us to dynamic range of a CCD. So what does it really means? And is there any differences between the CCD chips from one and another? It is clearly is. The size of the CCD chips determine how much electron can actually hold in the pixel well. So the dynamic range definition is in terms of CCD chips is the full well capacity divided by the camera noise related to the ability of a camera to record simultaneously very low signals alongside the bright signals. All right. So I know this is a bit mouthful and lengthy. What it means is full well capacity means the maximum number of electrons that can register a signal in a pixel. So the larger pixel give you higher well capacity, which also brings you higher sensitivity. Okay, larger signals also better signal to noise ratio and also higher dynamic range. So when the charge in the pixel over here, when you image it, when the charge of the pixel is exceeding the saturation level here, saturated pixel, the camera starts to deviate from a linear response that will compromise your quantitative performance of the camera. Next is excitation light source. This is very useful if you're doing fluorescent Western blotting. Here I'll take out two types of excitation light source, which are LED, light emitting diodes, and laser diode. LED is having a broadband light source. As you can see here, it has relatively low power output, diffuse light, but it is small and it comes with an economical price. Laser diode, on the other hand, is a monochromatic light source, higher output that delivers more energy to the sample, and it gives you premium performance as well. As you can see here, LED on the same plot will give you higher background noises, and laser diode will give you very clean background. Right. So here, laser excitation light source offers lower background, less light leakage indicated in blue here, and more sensitive in shorter time. Right. Here we go into the depth of fluorescence Western blot. We can divide fluorescence signals into two big categories: RGB, red, green, blue, and near-infrared fluorescence signals. RGB, visible fluorescence, we have Psi 2, Psi 3, Psi 5 dyes. And for near-infrared fluorescence, then we have IR 680, which these two are 700 nanometer wavelength and above. Right. So if you're doing fluorescence Western blotting, you can, just like you're doing chemiluminescence, you can opt for fluoro conjugated antibodies or fluorescence secondary antibodies that by considering the mix and match on hosts and species and dye. Right. So these are the pros and cons if you're between the RGB and near infrared fluorescence signals. RGB, it can allows you to do up to 3 multiplex target on the same image. It has wide dye application and dye choices. You can side dyes, GFP, RFP, but it has higher membrane and sample autofluorescence, thus higher background noise, and it has lower sensitivity when you compare to near-infrared fluorescence signals. In terms of near-infrared fluorescence signals, it only allows you to do 2 multiplex target due to the lesser dye choices that we have right now. Oh, sorry, this is a bit wrong here. This is good because near infrared will give you lower membrane autofluorescence signals, which in turn give you low background noises and higher sensitivity. Right. Finally, Western blot normalization. We have two major normalization method here: housekeeping protein and total protein stains. Housekeeping proteins is routinely used as a loading control. It relies on a single indicator of the sample loading across the entire membrane. We often choose highly expressed protein because we rely on the accuracy of this expression. We expect it to be stable and across every experimental condition and so on. But experiment has been shown that the expression of housekeeping protein has been varies across different experiment condition and treatment. For example, cell conferency, disease states, drug treatment, cell and tissue type, and so on and so forth. Thus, there's another techniques or normalization method that propose to overcome these drawbacks, which is total protein stains. How it does is it uses the combined signals with many sample protein bands in each lane. Thus, it will minimize the error and variability because you have larger samples to normalize right now. And it corrects the variation in terms of protein load. Because if you, let's say, I loaded lesser protein in lane A, I have then a total protein will show you that, oh, it has lesser, and you will normalize against within its the band of your interest. And it can be used to monitor protein transfer. However, it requires specific channels to capture signals based on the stains that you choose. Right. So here I want to say that total plotting is a new normal for Western blotting. We start to see this in publications because the advantages is outweigh the total protein, the housekeeping protein. Scientific communities, here I'm crop out Journal of Biological Chemistry and Proteomics. They have been proposing and stating the problem with housekeeping protein and saying that total protein stain or normalization will actually represent the true actual loading amount more accurately than the housekeeping protein. Right. Here, I'm using fluorescent Western blotting as an example of how to do housekeeping protein. You have a lot of proteins blot images over here. So you have definitely one of them as the image, your housekeeping proteins. So this single housekeeping protein, after you capture, you analyze it, get the intensity, signal intensity of each band, and also in terms of volume and a certain value. Then you use the value to normalize against, let's say, this band over here, the protein of interest. So in terms of fluorescent Western blot, it's the same as chemiluminescence. The methods are all the same. Right, next is total protein stain. So this is what I mean about total many bands, protein bands in a single lane, and they serve as a total protein normalization. So all the bands in that stain will be used calculating the normalization factor, which is thus it gives you more accurate more accuracy in terms of total protein normalization in the Western blotting. Right. Okay. Right. I say that. So this is a graph showing you that total protein stains. Here we have Azure red that stains for the total protein. Just want to tell you that it has a higher signal linearity when you compare to, let's say, actin, tubulin, GAPDH, which is proven in this plot here, when you increase the protein load. Right. So let's recap what we have talked about today. First thing is the principle of Western blot. Second is antibody detection with chemiluminescence and fluorescence method, and how you should choose between them, or you use both of them to answer your biological question. Next is imaging Western blotting. There are a few things to be aware of, like the CCD chips, dynamic range, X-ray film limitations, and also the advantages and excitation light sources if you're doing fluorescence Western blotting, and also RGB and near-infrared fluorescence dyes. Finally, normalization we are talking about housekeeping protein normalization and total protein normalization. Right. Thank you for your time today. Azure Biosystems is here to fulfill your Western blotting needs. If you're looking for Western blot reagent, imaging system for your lab and setting up a new lab, we have a range of gel system and reagent for your Western blotting needs, such as a tank, aqua gel tank, Radiance, which is an easier substrate, and Sapphire biomolecular imaging and Azure imaging system. Right, so here I want to say that we have Azure imaging systems that allows you to do near infrared Western blotting, chemiluminescence, which is something very common, chemiluminescence with color marker, free color fluorescent Western blot, bioluminescence bacteria, protein gel, Western blot that stains with the total protein stains of Azure red, DNA gel, and also finally fourth color fluorescence Western blot. We have 6 models over here. As you can see, when the number increases, so we have more modules included in the machine, in the system, and all these systems are upgradable on field, and yeah, in the same machine. You do not need to worry about returning the unit and get one unit. No, everything can be upgraded on field and can be upgraded as long as you want it. Right. So if that's a wrap for our webinar today. If you are new to Western Blot or you want to know what is the new technology or brush up the techniques or troubleshooting your Western Blot, please feel free to download our Western Blotting guidebooks at the link displayed over here, www.azurebiosystems.com/guidebook. Here, this guidebook encompasses both fluorescence and chemiluminescence Western blotting, covering every single details that you should know in Western blotting as well.